Okay, in this video we're going to discuss lineage commitment and what it is and the different ways or the studies they have on it. So lineage commitment is it's a positive thymocyte that must decide whether to join. It can decide whether to join two different ways. It can either decide to join the CD8 plus cytotoxic T cell lineage, or it can decide to do the CD4 plus helper T cell lineage. And so that's initially what lineage commitment is. That is the definition of it. And so lineage commitment requires two different things, and if it has these two things, then that can result in one of two different things, which we'll get to after I describe the first one. So lineage commitment requires genomic organization and gene expression. So if you have these two things, then it can result in one of two different things. So it will result in the silencing of receptor genes, which will make more sense once we get to how it works. And I'll show the diagrams, and that'll make more sense that we would want the silencing of gene expression. And the other thing it could result in is the expression of genes associated with a certain lineage function. So as long as you have those two things, the genomic organization and the gene expression, then you can get one of those two things. And then that's how that's how lineage commitment works. So there are three different types of lineage commitment. Not really types. So let me say this first. It is still a debate about how these cellular and molecular molecules are responsible for this cell lineage. So we have, what I'm about to show you are current perspectives or current studies, or I guess you could say theories on how this works, but it's still a debate, so none of them are for sure. I mean, they have proof that a lot of it's happening, but so we're going to talk about three, but really I'm just going to put three up here because we're going to talk about four, but one of them kind of just leads to the last one we're going to talk about. So the first one we're going to talk about is instructive lineage commitment, and we'll see that this one and the next one, which is and I don't know if I say it right, stochastic lineage commitment. We're going to talk about how these inevitably are very small. Studies are very small. They have very small evidence that, so then we end up still wanting to study on it. So then the three is the kinetic signaling, and it's kind of our most current one. But we're going to talk about one in between this 2 and 3, but it'll kind of lead to this kinetic signaling because it also doesn't have that much evidence, has a small amount of evidence, so we end up studying another one. So we're going to start, and we're going to talk about instructive first. And so this picture I'm going to show you has the instructive and the stochastic on it, but we mainly want to look at the instructive until I get to stochastic, but it's going to kind of compare them. I have two pictures on two separate slides. So as it's talked about, we're going to talk about instructive cell lineage, or instructive line commitment, which, so on the instructive one, the TCR, let me write it down so it'll make more sense. So the TCR and the CD4 and the TCR and the CD8. So together, so these co-arrangements, so the TCR and the CD4, these co-arrangements, these can produce rare, or if you want to say unique signals that initiate distinct development programs that are going to do this lineage commitment. So what would be involved in this lineage commitment is TCR, CD4, and TCR and CD8, which is in all lineage commitments, but that's how they would be paired in this one. And so this first picture is going to show you an immature lineage, or this is the immature on this side, and this is the mature on this side. So we're not going to look at the stochastic. So, and then you see that Well, actually, it looks like we are going to have to look at the stochastic. So it looks like, oh no, can you see the difference in them? Okay, so the dark bubble is going to be the CD4 commitments, and the light bubble is going to be the CD8 commitments. So, and then this one is uncommitted. So when they're immature, they're uncommitted. And we get over here. So after the instructive ones, we get, it looks like double the CD4s that we do the CD8s, and then down here it looks like it's all CD4s. So this is with instructive. This is the instructive model. So I guess an example of it would be from looking at it, and now we'll go to the next one. This kind of again compares them. So and you see what you get here. So and this one, the pink is the CD4, and then the square is the CD8. So this one kind of shows more. So the square is the CD8, and that one's it. So an example of the instructive model would be if a thymocyte randomly generated a TCR with an affinity for the MH class 1, which is kind of what we're showing here. So this is the MH class 1 is going to be the open square. So it's right here, the CD8. So if that happened, then the TCR and the CD8 would bind the MHC class 1 together, which we see happening here. And then, and as well as it would generate a signal that would specifically initiate a program that would silence the CD4 expression, and then it would induce expression of genes specific for cytotoxic T-cell lineage function. So, and then we see that here, and that's what I explained. So the TCR and the CD4 and the TCR and the CD8, they generate these signals that develop a program, and then in turn it'll silence the one that they're not working with. So in the example I gave, it would silence the CD4 because we're working with the CD8 and the MHC class 1. So this would, so if you were doing the opposite way and it was the TCR and the CD4 co-engagement, they would generate a signal that would silence the CD8. So it's just the opposite. So it's kind of a hard concept to understand, but it makes more sense once you look at diagrams. I had to watch a couple YouTube videos, Khan Academy, to kind of make it make sense, and it's hard to understand just looking at things like that, but I guess reading it and putting it to a picture helped. So now we're going to go to the next one, which is the stochastic, if I'm even saying that right, and we'll go back to our first picture so we can look. And we see in the immature ones, so they are already separating the immature ones, unlike in the instructive ones. And in the stochastic one, it's positively selected thymocytes that are randomly downregulate the CD4 and the C8 places. So, and that's what's going to happen in these. And this one has a lot more concepts to it. It's a little more complicated than that. The TCR and the CD4, like I stated, just written just like this, the way we wrote it the first way. And the TCR and the CD8, their co-engagement does not necessarily generate distinct signals. So that's how it's different from the instructive, is when just because they're co-engaging, like I said, this way doesn't generate distinct signals like it did in the instructive. Because if we had the TCR and the CD4, then we would be getting a signal for the silencing of CD8. And vice versa, but that doesn't necessarily happen in the stochastic one. So we're going to go to our next one, and we see how it's a little more complicated. So the one that can co-engage with, okay, so only the cells that express the right the core receptor generate a TCR signal strong enough to survive to mature. So for them to be the correct receptor, it has to be the one that can interact with MHC with the TCR as well. So like I said, it doesn't necessarily generate distinct signals, but it is a little more complicated. But it can produce the same signals, but in most situations it does not produce most signals. And like I said, the difference is it has to have that correct core receptor, and that correct core receptor has to generate TCR. It'll generate a TCR signal strong enough to survive to mature, and that would be what we have down here. So, and like I said, they these are still debates about these, and so so like I said, there was not enough evidence to prove, or there was evidence, but it was small evidence, and they were very similar. So a lot of the same evidence supported both of them, so they couldn't really decide. So then, like I said, we move on, and we're going to go to the kinetic signaling, which is a little bit different, and I think I have a picture of it if I put it on here. Yeah, so it's a little bit different. It's our most current model, and it says that thymocytes commit to the CD8 lineage if the TCR signal is interrupted, which in my which makes a little more sense to me than the other one. So here's our TCR, and then it's interrupted, so then it's going to bind to, or it's going to commit to that CD8. This model, this kinetic model, it has, it took learnings from our first two models, and it put it with what they currently understood at that time, and it just added them together, so it makes more sense. So, and I'm not good at explaining how this works, but I can try. So by stopping that TCR signal, or like I said, by interrupting it, you can make the cells that normally would commit to the CD4 lineage commit to the CD8 lineage, or the CD8 cells, and then vice versa. If you instead of inhibiting that TCR signal, if you enhance it, then the ones that were going to commit to that CD8, to the MHC class 1 cells that were going to commit to that CD8 are going to commit to that CD4. So, it's still kind of the same concept as our instructive one, but it's just a little different because it's talking about you mess with that TCR signal, which makes a little more sense to me. So our three proposed models of our lineage commitment were instructive, stochastic, and kinetic signaling.